Okay, I'm just going to wait a minute for the people to jump on. Okay, good morning, or for some of you, good afternoon. My name is Bill Coakley from BSMA. I want to welcome all of you to our first webinar of the spring since our annual conference um, back in March. Um, I wanted to go over a couple instructions about the webinar for all of you. The first is to let you know that this event is being recorded. So anybody who registered for this event will be receiving a copy of the recording shortly after the webinar. Second, there will be time set aside both during and at the end of the webinar for questions. If you do have a question, please use the Q&A box as opposed to the raising hand. And the chat box has been disabled. So again, you enter any questions you have anytime during the call in the Q&A, and at the appropriate time, that question will be fielded by our moderator and directed to one or both of our panelists. So with that said, I want to welcome Catalent and hand the event over to Bonnie Moore, who's the Vice President of Clinical Supply at Catalent. Thank you.
Thank you, Bill. On behalf of Catalent, we appreciate the opportunity to kick off the spring webinar here today. So as Bill said, my name is Bonnie Moore. I'm the Vice President for Catalent Supply, Clinical Supply at Catalent. And I'm here to serve today as the moderator for our webinar titled Effective and Flexible Packaging, Labeling and Distribution Strategies for Diverse Drug Types. We're honored to have two distinguished speakers with us today. Kate Carey, who serves as our Director of CSS Project Excellence at Catalent, and Marcel Vitek, our Global Project Manager at Catalent. During this webinar, Kate and Marcel will review the latest advancements in flexible packaging, labeling, and distribution strategies. They will share insights on how these innovations can enhance clinical supply chain effectiveness and address the unique challenges faced by sponsors in the biopharma industry. As Bill has explained, we encourage you to actively participate by asking questions throughout the presentation. We do ask that you use the Q&A function. I will be monitoring those throughout the webinar today, and we have set aside time at the end to address those. And lastly, thank you to BSMA for providing us this opportunity to present to you all today. So without further ado, let's dive into today's topic. Kate and Marcel, the floor is yours.
Okay. Thank you, everyone, and thank you, Bonnie, for the introduction. So I will give a brief introduction about myself. So I'm Marcel Vitek. I have been working in the pharmaceutical industry now for around 10 years, and all of those 10 years were actually at Catalent. I worked in several operational and also project management related functions, mainly in Catalent's clinical supply service business. And I'm currently a program manager with a focus on strategic supply solutions. I'm based out of Germany, which you probably hear from my accent, but actually I spent most of my time and in my career in Japan. So that makes me also an expert in clinical supply services in Japan. My educational background is in chemistry, and I hold a PhD in organic chemistry from the University of Heidelberg in Germany. And now I hand it over to Kate.
Marcel. My name's Kate Carey. As Bonnie said, I'm Director of CSS Process Excellence. I also come from a background within Catalent, 20 plus years of experience in operational and project management roles. And looking forward to talking about this webinar about different strategies for packaging. So we'll get started. Our agenda today is we're going to look at different supply models and how they were developed and why they were developed. And then we're going to also talk about different attributes and benefits of the demand-led model, which is going to be the focus of this workshop. And moving on to what we call the hybrid model, which and what additional benefits that model provides on top of the demand-led. And then we're going to go over some key process and systems and tools that we use to deploy the demand-led service. And then at the very end, we're going to do an exercise with a workbook and walk through some kind of real life examples. And then at the end, we'll do the wrap up and any additional questions from the team. So we will go to the next slide and start going into the detail. We'll give you some background. So why were these different models developed? And what are those drivers? So one of the big things in recent years is there's a drive to get to the market quicker. There's a speed to start studies and be first to market. In addition, we've seen a more green sustainability perspective out in the market. So there's a want to reduce waste, use materials more effectively, and have sustainable logistics requirements around study development. In addition, there is different product differentiations, like adaptive trials have become more prevalent. And along with that, there's an increase in the need or in the research around biologics and ATMPs, which goes into the next thing, increased product costs. When we talk about biologics and advanced therapies, they have a tendency to be a little bit higher in manufacturing costs, which has driven the need to be effective in use of material. And then the last 2 points are similar in that there is a greater need for patient and clinic centrality, and then also moving to some requirements around personal medication, such in the gene therapy space. And so those are the drivers of why we need something more, a different model to approach these types of drivers.
Okay, thank you, Kate. And now I will going to present you the 3 different supply models which we are currently using for packaging and also sending clinical trial material to investigator sites. So on the top, you can see we have the so-called traditional supply, which is a centralized supply from typically one location. Then there is just-in-time, also known as GIT or JIT. And also then the last one, this is the main topic for today, is the demand-led supply, which is a decentralized approach. Okay, you can go to the next slide. On this slide, you can see an overview of the supply chain from the drug product manufacturer via the primary and secondary packaging steps until the distribution to the investigator sites for the patient visits. In the top, you can see the traditional supply, which we also call a push model. We call it a push model because it's just in case packaging with a high overage production in order to mitigate any inventory risks. So large batch sizes are packaged up front and then stored at one packaging hub. And then the material is sent to the clinical sites where the stock is then held in anticipation of a predicted demand. Whereas for demand-led approach, it is a patient-specific packaging. So when there's an actual demand, for example, when a patient enters the screening, then the material is packaged and then distributed to the clinical site for a known demand. And this is why we call it a pull model, because the patient entering the study and there's actual demand for shipment. So it's a pull for starting our packaging. And Here we have now smaller batch sizes because it's from an actual demand. And the packaging here typically takes place at regional packaging hubs. So for example, if there are clinical sites in the European Union, then we would do the packaging in Europe. If there's an America region, then we would do the packaging in the US. But we also have country-specific packaging in Japan or in China, for example. This brings then the packaging close to where the actual need is, so where the patient is, and reduces the turnaround times for the packaging and then also the distribution to the clinical sites. So for demand-led, we have now two different packaging models. One is pack to stock, or in short, PTS, which you can see in the middle. And then there's also pack to order, in short, with PTO, which is shown in the bottom part. In both cases, we have bright stock, which is unlabeled bulk material. Both is packaged for an actual demand. The difference here is that for pack to stock, we do the packaging for a specific time period. So we have an actual demand for, let's say, the next couple of months, so let's say 3 months. And then we do the packaging on a frequent packaging basis for those 3 months to cover those actual demand. And then we store it at our packaging hubs and then send it to the investigator sites as per IRT order requests. For pack to order, which is a true demand packaging, it's a little bit different. So when the patient visit is planned, then the IRT orders are raised, and this is the trigger for us to start the packaging. So the batch sizes here are even smaller than for pack to stock. And the material in this case is not stored intermediate at our packaging hub, as we send it directly to the clinical sites after packaging completion and after the final release, of course. So this should give you now an overview about the supply chain for those two models. And on the next page, I show you the difference in some aspects between traditional supply model, demand-led, and as well just in time. For the traditional model, the kit assembly is done in large secondary packaging campaigns based on an assumed enrollment rate using either single panel labels or booklet labels, so both is possible. And it's a full batch record and a full release at the end, and we consider this as one packaging step. For demand-led on the right, in this case, it's for pack to order. The kit assembly and label application is done prior to shipment based on the actual demand. And here we usually apply country-specific single panel labels. And also we have here a batch record, same as for the traditional supply, and also a full release prior to the shipment. And we also consider this as one-step packaging. The difference now to GAT is that for GAT, there's first of all, it's a two-step process. So it's consisting of a base label application as a first step, which is equivalent to traditional packaging. And then it's followed by application of an auxiliary label in the second step, which is the actual just-in-time labeling. But this label is only applicable for the outer part of the kit. So it would be, for example, on the kit, but it would not be on the inner parts like a vial or a syringe. And here also for GAT, it's important to mention that there is no batch record. And packaging is performed in a warehouse where the distribution takes place. Whereas, and this is the big difference to demand-led. In demand-led, we have a batch record and we have a full release and we do the packaging in a secondary packaging room. So this should give you now a summary of the differences between those three models. And I hand it over to Kate.
Sorry, I went a little fast there. So what are the attributes and benefits for the demand-led model? So some of the attributes, it's decentralized, as Marcel explained in the previous slides, where we have regional facilities that can help support these packaging activities. We manufacture closer to the patient, meaning the demand that's triggered from the IRT or shipment request. We have global standardizations again around the network of facilities that are supporting this. One of the big ones is the ability to pool supplies across multiple protocols. If you're using a particular compound across multiple studies and trying to use limited drug product, this really supports that pooling of material, and we've seen real great success around that. Also, one of the attributes is we can use single panel or label groups. We can also use booklets, but the really driver for efficiency and speed is single panel country labels. We do order aggregation, which we'll go into a little bit more detail in later slides. And then we use that system integration so that entering the data once and it flowing through the process on our side. Some of the benefits, the quicker lead time for new studies if you're not using a booklet, or if you're pulling supplies and you have some of the master labels designed so that you can pull, you can start new studies pretty quickly. We can add new countries with single panels instead of updating a booklet with adding another page for another country and doing another large run. You're able to just add a country, and you can design for a particular country and then never use it. So it's efficiency of activity. Overall, it can reduce overage, it can reduce storage costs, and it can also reduce shipping costs. Again, because you're closer to what is actually being requested, you limit what is actually being used and consumed for that study. If you have a particular design where you are working to have greater patient compliance in that design and how we supply the material to the sites. And then there is a much easier expiry date management. Instead of labeling material up front in a booklet and then hitting an expiry date and doing retest date labeling, we have a process where as stability data comes in and you need to update that expiration date, we update our master documentation, and when we do the next packaging run after that update, we are using the most up-to-date expiry for that next label print run and assembly of kits. Also, we can do patient-specific manufacturing and patient-specific labeling. So as an example, in the gene therapy space, if there's a requirement to have a kit that has 10 vials for one patient and maybe 5 vials for another, if there's a need to have any kind of patient-centric information around that, we can print that on the label because we're doing it in real time based off of a request that's specific for that patient. And if they need fewer kits based off of weight or more, each run can be customized to that specific requirement. So what is the hybrid model and what additional benefits does it provide? So the hybrid model combines the demand-led pack to order or pack to stock along with the traditional supply model to cover different phases of a particular study. As an example, in the initial phase, during enrollment, the demand, as we probably all know sitting here on this webinar, can be unknown. It can vary. And it also changes, right? One set of countries ends up not going live and you have to adjust it and add countries or add sites in other regions. So it becomes basically a little bit more on the unpredictable side. So during that time period, demand-led is a really good option to fulfill that unpredictable demand where you're not allocating based off of a estimate, you're allocating based off of a more real piece of data, either the shipment request or a shortened time period if you're using pack to stock. Then when you move to the maintenance phase, when enrollment is almost complete or complete, your demand is more known, constant, fixed, and predictable. At that point, moving to larger batch sizes where the packaging activities are done further apart and potentially even going to a booklet label at that point for a particular program, that's where the traditional supply model can be used in conjunction with the demand-led. And that gives you the ability to have more flexibility throughout a particular set of timeframes. So this chart here kind of displays the how the demand looks between these different models. So the white line there is the traditional model where at the beginning of the study, you pack a significant amount of material and then you consume it and then you pack another. And you do that throughout the study. The dark blue line is the demand led, where you're doing this example in this line is this packed order. So you're only packaging what you're consuming essentially. And then the hybrid model in this example is a combination of pack to stock and then moving to a larger batch size traditional model at the end of it. You're only packing small quantities throughout the study based off of what your viewpoint is for patient demand. And then when you get to, in this example, around week 20, you're deciding to pack larger quantities. And then knowing what that demand is allows you to have that less packaging based off of known demand. So going to the next slide.
Okay, that's me. Thank you, Kate. So what are now some of the key processes, systems, and tools which enable a demand-led model? So first, we have, of course, a global standardization across our decentralized network. So all our hubs are using the same processes, the same systems, the same documentation and templates, so that we are all aligned in what we are doing. Then, of course, an important point is the order aggregation. This allows us to aggregate the incoming IRT orders into fewer packaging ones. This reduces the number of setup steps, line clearances, batch record, review, and release steps. But I will show you later more details on how the aggregation works. Another key aspect is the barcode traceability. So our bright stock is controlled by a 2D barcode. This can be either inkjetted on the primary package material, or we can apply a small 2D barcode label on it. This allows the control of the material through to the clinical finished goods. An important point is also the system integration to avoid having multiple entries in various systems. Just an example, our IRT systems are integrated with our aggregator to allow an efficient and safe way of creating of the packaging orders. And of course, also we have a dedicated team collaboration between all involved parties. So on the next slide, I will explain you now how the aggregation works. So the purpose of the aggregation is to gain packaging and QA release efficiencies. So without the aggregation, we would have a lot of batch records to create and QA releases, as there would be a lot of IRT orders coming in. So this is where the aggregation reduces the amount of batch records and QA releases significantly. As I said before, we have system integrations between various IRT vendors and our aggregator. In the example below, we have a very simple clinical study with only two different pack types, one active and a matching placebo. There are three different shipment orders, one with two placebos and one active, one with one placebo and one active, and the other one with three placebo and one active. So the aggregator will now combine those 6 placebo kits into one assembly order, which is our name for a packaging order. And the same also goes for the active, where we have now 3 active kits packaged in another assembly order. The placebo and active kits are, after packaging completion, put into 3 different inner shipper boxes for the distribution for the 3 different site shipments. We also put the activated temperature loggers in it and close the cartons. And after the final QA and QP release, these shipments are ready to dispatch to the clinical sites. Our standard turnaround times from receiving the IRT order until being ready to dispatch is as low as 3 working days. Okay, now back to Kate.
Okay, thank you, Marcel. So the next step is our exercise. So we'll pause here to see if there's any questions, Bonnie.
Yeah, Kate, we have one question. Thank you, Marcos, for your question. It's a question that we get frequently and has high variability, but I think Kate's going to try and address it for us. And just for the attendees, the question is, how do the per unit production costs differ across these models?
Yeah, as Bonnie said, it can change. It can be dynamic depending on several factors. But from a general perspective, the per kit price on the full pool model, the pack to order, is higher because you're doing more packaging activities, and it is a quicker turnaround time. And it gets less expensive as you go back on amount that you're doing. So the pack to stock's a little less compared to traditional. Now the savings in the calculation we walk people through is, what are you saving in other areas, such as manufacturing batches. You may save on not needing as much. And it's a good question going into the work, kind of the next step, the exercise we're going to walk through, because we're going to demonstrate that difference and where the savings is seen. So maybe hold off on that. And if there's further questions when we get through the exercise, that may help answer part of the question, but the actual cost of packaging is slightly higher on the pack to order. Were there any others, Bonnie?
Yes, we do.
Okay.
So there's one here. We have two more around QP release, how we manage QP release with the model, and then one around IRT info received in real time. So maybe Kate, maybe I suggest we walk through the exercise
and then come back to them. Okay, just to make sure we have time.
Absolutely.
Okay, so we will, let me just get myself back here. So we're going to jump to the exercise. I'm going to go through some of the information to give us a baseline before I go into the spreadsheet. So we're going to look at two different studies that are using the same IMP and comparator product under a program of work. And they're starting within 3 months of each other. We're going to kind of, because this is a webinar, we're going to assume some things and to build that supply strategy for the first packaging set of campaigns. And we're going to look at conventional, traditional, and then we'll look at decentralized, the demand-led version of those if we were going to pack for these. And then we are going to essentially take what real information on some assumptions of how that study would have actually run versus what the plan was at the 12-month point. And so we'll look at how what primary pack material was utilized versus what was actually needed. And we'll compare the two models at the very end. So I'll give you a little bit more detail about the studies here. So here's some information about the studies. I'll go over the highlights, right? It's a double-blind, double-dummy parallel studies. They're very similar. They're using, in the detail, there's 4 treatment groups, but we're going to focus on the active. So basically the pack type 1 and pack type 3, the IMP active and the comparator active. There are similar cycles and window treatments. The big difference between the two studies is one has essentially higher number of patients, which means more countries, more sites. And the first study is starting in May of 2025 in this example, and the second one starting in August, 3 months later. And the second one, I think, runs a little bit longer. It's a longer number of months in total. So that they're very similar, basically, with slight differences. And then as far as the drug products, just some highlights on the 2 different strengths that we're going to focus on, the IMP and comparator, and they both have expiry dates of September 2026. And then our enrollment assumption here is the straight line enrollment. As you can see, it's for the general, it's starting in May and then ending for both studies in the 2029 timeframe. So I'm going to stop sharing the slideshow here, and we should, let's see, did I lose? Can you guys still see my screen?
Yeah.
Or did I lose it? Yeah. The spreadsheet?
Yes.
Okay, good. Thank you. Okay, so some information that we're going to go over. So for packaging centralized in year one, we're going to assume for this activity that we're walking through that we're looking at the first 12 months. So this is study one and study two. In our model, if we had a different avenue, we could change this and modify it. But for the sake of the time frame we have, we're going to assume 12 months. Assuming this, our supplies can last till, for study one, April 2026, and then we need supplies to last till July 2026. And then this calculation is just saying if we assume 12 based off of what we have, the comparator is still good based off of the expiry of September 26. We're going to assume in this activity that we have a 30% overage. Depending on the organization, this can vary, right? Some organizations use less, some do a lot more overage. But for the sake of this, we'll do 30%. So at 12 months, the planned straight line enrollment, we would have for study one, 93 patients enrolled based off of the curve. And for study two, 165 patients. So that would assume, based off of us doing 30% overage at 12 months, that we would need to have packed for study one around 2,000 of the IP for that study, and over 4,000 for the IP on study two, and then for the comparator, around 1,000, and then 2,000 for study two. So we're upwards we've packaged per our plan 6,442 IP vials for the two studies, and then 3,221. We do have enough material because we have 6,000 of the treatment one. This looks like it's off, but that's less of the maybe concern right now. So that's centralized. That's what we would have needed if we said we're going to pack up front for 12 months. That's what we would have packed. So what would that look like if we were doing a decentralized demand lead? We're going to assume the same 12 months. And we are going to do single panel. We'll assume single panel. And instead of 30% overage, because we're closer to that patient demand, the loss is if you have dropped patients or you have material that you sent to the site that the site doesn't enroll, but if you wait till screening, you could limit that. So 5% is what we're going to use for this workshop. So based off of what we will assume is an actual patient enrollment, which we have calculated in the background of the spreadsheet, is the actual at 12 months was 77 patients compared to, I'll just flip back real quick, what we assumed is 93. And the assumption at 12 based off the straight was 165. And we only had 106, right? So there is a difference in actual versus planned. And because we had less overage, also, we're going to need less kits. So for study one, the IP was 1,000. Study two was 1,800. And then for comparator, we had about 500. Study one and just under 1,000 for study two. So that left us with only needing 2,000, just under 3,000, 2,920 of IP and 1,460 for the comparator. So we can see there's a difference there. Now, if I change this, let's just say for the sake of conversation that I change this to 8% or something, right? That changes it. We'll leave it at 5 and we could go back and look at it a little bit more at the end if we have a little bit of time. So we'll leave it at 5% overage. And this is where we'll do the full comparison of what is the difference and how did that affect when we use the different models. So for study one, we added some countries that weren't planned in this activity that we're walking through. We actually had 20% screen fail rate for study one, which was 15% more than the planned. And then we had 15% for study two, which was 10% more than what was planned. Our target enrollment, like we reviewed before, was 93 and 165, and we actually enrolled 77 and 106 at the 12-month mark. So the enrollment plan versus actual was, it was slower to start recruitment in study one, and then study two, just they didn't recruit as quickly as they expected. So we're going to look at the central and then the decentralized, so traditional and demand-led. So for study one, we packed, like, we'll go back and review what we packed. We packed 2,000, a little over 2,000. We only used 1,000, which will be similar to both these. So we had a remaining of almost basically double the amount of kits remained from what we packed to what we used. And this similar story, actually a higher percentage of what was not used for study two. This is actually study two, sorry, right here, study two. Make this bigger. So we had, for the IP specifically, we had 3,661 remaining kits unused that we packed, that we put a clinical label on, that if we wanted to use anywhere else, we'd have to repack in some fashion, or, right, you just have to do expiry updates if you hit the expiry date. So that's a waste of almost $5,000, a little over $5,000 on the IP. If we look at the comparator, similar story. We packed a little over 1,000 and a little over 2,000 each study. Kits remaining for study one was 500, kits used was 524 and 867. So in total of unused remaining kits were 1831, which is a $10,000 cost. The cost here for the IP is 1.5 K per vial. So that puts a waste under conventional of 16. So if we look at decentralized, with our screen rail fate and the 5% overage, we had only between both studies 139 kits that were not used. So that was only $209 worth of waste, of $209,000 worth of waste. And then for comparator, we only had 70 remaining. So that was only $417,000. So we compare that to 626,000 compared to 16 mil. It's a big difference. This is where, right, kind of to the question earlier, is your packaging costs might have been higher, but you effectively used your product much more efficiently. And that, unpackaged material could have been used for study 4, 5, and 6. I'll kind of pause there to see if there were any additional questions that we think would be good to answer.
Yeah, Kate, we've got a couple of other questions that have come in. So while we're on sort of this cost model, Kate and Marcel, can you tackle the difference of the order lead time between a conventional model and a decentralized model?
I'm assuming it's about the timelines around packaging activities. So the decentralized model, I think Marcel kind of mentioned it in the presentation. For pack to order, it can be as short as 3 days. Those 3 days assume some, and there's probably some questions around quality release and QP release. That assumes that a client is either committing to a very short post review and is going to review all those records, or they've qualified Catalent to do that release on their behalf, and we're able to meet that 3 days. And we're actively doing this for studies currently. So it can be as short as 3 days. If a client is reviewing those, it might be slightly longer. And that's from receipt of order to dispatch out. There's additional time for transit from Catalent through the courier to the clinical site. And that's obviously country dependent, site dependent. And then for pack to stock, we can do that. Typically what we say is that's around 10 days. So that's from order receipt, so that quantity of material that we're going to ship, to released, ready to receive a shipment request. So the time frame is a little bit different, but it's around 10 days. And that's kind of batch size dependent, that usually most of the batches are within that achievable time frame.
Okay, great. Thank you, Kate. We do have one other question. Marcel, I'm not sure if you want to speak quickly. Kate's addressed it high level here around managing the QP release.
Yes, so as Kate mentioned, so there are two ways. So typically when we have pack to order with those 3-day turnaround times, the client authorizes us to do the QP release. So our QP team, they typically do it then on the last day, on the day 3, the QP release, so that we are ready to ship. So this has never been an issue, and we have standardized batch records so that it's very smooth and quick, the QP release.
Great. And then one of the other questions that I think is really important to answer for the attendees here today is, Kate, Marcel, can you speak to the number of or percent of clients that are using this model, and maybe some of the benefits that feedback that we get from the clients using this model who previously used a conventional traditional model and now have moved to either pack to order, pack to stock, or even are moving now into the hybrid model?
Yeah, so we have some great examples of use of the hybrid model. So we had, let's say, a early adopter use pack to order on some studies where they had challenging drug supply and needed to pool material quite, to make the difference in them being able to start we're talking multiple studies at the same time. So what they've seen and achieved through using the hybrid model is, for the studies that have become, even the products that have become more developed in their system, longer expiry dates, and also seeing the studies get to a point where enrollment is met, they've moved to using our pack to stock model to do larger batch sizes. So they've seen the big benefit we've seen a lot is the pooling of supplies. If you're in a position where you're trying to use drug product and lots across multiple studies, this allows the efficiency of starting studies and not committing all this inventory to study 1, study 2, study 3. It's uncommitted until that order comes in, and you're able to start more studies in a shorter time period. So that's part of the benefit that we've seen on that. As far as the amount of adoption, we've seen that even within the last year, we've seen much more interest and more people thinking, right, this makes sense especially if they're trying to be aggressive on drug development on its particular compounds.
Great. Thank you, Kate. I think we have maybe one other question here around the top 3 reasons for waste of inventory. I think you covered it a little earlier, but it might be helpful to walk back through that.
Yeah, so I think somewhat of this activity kind of addresses it, right? So we assume a certain quantity of overage. I mean, 30 could be on average if you did even more overage because you're unsure of like where the demand is or how quickly you're going to recruit. That's where we see a lot of waste. The other piece is when you have a drug that's early in development and you have short expiry on it, you could have a situation where you've labeled it, you're trying to use it for a study, and you're consistently needing to update it with new expiries. And you, in some instances, you get to a point where you're kind of limited on another expiration date, even on the physical label. So it becomes a difficulty of supply. And you may just have to set that aside and rethink that and go to your next lot if you're in that kind of situation where the demand led because we're able to either package much smaller quantities and pack to stock or only use what is needed. Is you're only touching that product once before it goes to the clinical site and to the patient. So you're getting the most up-to-date expiry. I don't know, Marcel, if you had any other things to maybe point out that I'm not thinking of.
No, you
on
covered
wastage.
And Marcel, Kate, from an overage standpoint, in the demand-led model, you're truly packing to order, right?
Correct.
So you wouldn't recommend any overage in a true pack to order model. Do you have any recommendations or any guidance or experience that you're seeing from clients as they're moving towards the hybrid model on or pack to stock model? Any levels of overages that you're seeing teams use? You know, typically we can see anywhere from, I've seen 30% to 200% in overage, right? But as you move towards that hybrid model or pack to stock model, any guidance on overage that you would suggest there?
So on the pack to stock model, as you can see kind of in this example, it really it could be pretty low. It depends on right, in this example, the light blue, you're doing it it looks like almost every month, you're doing a packaging activity or resupply. So your risk around that and the potential 10-day turnaround, your overage could be very small because if you planned it wrong and you're from a clinical supplies management perspective, kind of keeping an eye on the IRT and the consumption. If you say, oh, I planned and didn't have enough overage, you just trigger another activity, right? So, and you're getting it within 10 days to be able to release it into the IRT and have shipments then drawn off of that new run. So that it can be very low. The overage for pack to order is from just the packaging activity, right, is almost zero. Your overage in consumption loss is more it got to the site, a patient dropped, and that site didn't get another replacement patient or something like that. So it's really more the material left at the site that's unused or at the end of a study, right? You triggered that the levels in the IRTs, the floor and the high. So right, if your IRT settings are maybe conservative, you may have shipped more to the site than you actually end up using, depending on patient visits and drops, screen fails and drops from the studies. So it's almost zero from our side on the packaging. It's more what's left at the sites overage.
And then I'll just remind the participants, I think we have about 9 more minutes left, but happy to answer any other questions that you have if you would just put them in the Q&A chat. Marcel and Kay, have you experienced or any feedback from any sponsors? Are there any downsides to adopting the demand-led or hybrid model?
Oh, let me think. The downsides. This is not a typical Bonnie, you got me there. The downsides might be, right, if your organization is not ready for that change, right? Some of what we talked about earlier around reviewing of your supply very differently. Your IRT setup pieces, if you haven't really thought through that and made adjustments for that, and expectations of your clinical team, because if you have a 3-day window of us packaging along with transport compared to a standard of 24 to 48 hours of Catalent processing time with transport, it's a shift change, because they do have to wait a little bit longer from order raised to material at site, and you have to make adjustments. And then around the quality release pieces of it, you have more batches that are in process for both the pack to order and the pack to stock. And if you haven't accounted for how do you sustain that, that can be a negative. It's more of, if you haven't planned for that adjustment of thought and how that changes your organizational standards, then it could be seen as a negative. But if you've planned for it, and we walk people through it, right? We're trying to set you up for success if you go down this path. We want to make sure people are using it for the right kind of studies. Not every study meets what we would recommend this for, because there's a higher cost to the packaging activity. That's maybe the other, I don't want to say it's negative, it can be perceived as negative. It's just a factor you have to account, because it might be, if your cost of product, like we said for the example, if I can get it back up here, if the product is more expensive than the packaging cost, may be higher, but you've saved, significant money on drug usage, and you've avoided maybe a comparator purchase. You've reduced your comparator need, or you've potentially saved a manufacturing run, because you didn't need to go into a third, fourth manufacturing run, because you're able to use that product more efficiently for the studies that you had in your pipeline.
Thank you, Kate. We've got a couple of other questions that came in, so I'm going to try and grab a couple of them quickly here. Is there any difference in the bulk material that you would hold on site available for packaging and labeling across these different models?
Is there a difference in the bulk material? There is a difference in the sense of everything is held as unlabeled for the pack to order model. And there's some differences in how you view that material in IRT. It's essentially virtual inventory allocated to that study. But you do, right, so maybe the question is more around the regional decentralized. So if you're packing centrally, you probably have your drug lots in one location that you're going to put in a booklet label, and it's all sitting there. In the decentralized model, you're putting it at regional hubs. So you do have to split your bulk material. Let's say some sitting in a US region to support US, Canada, and maybe South America. And then you have some of that unlabeled bulk material sitting in EU, as an example, that's going to support EU and other, European non-EU countries within that region. I think that's probably what the question was.
Yeah, I think so. Okay. I think we've got 4 minutes here, so I'm gonna throw one other one at the team here. I think you've spoke about it previously, but maybe you can just dive a little bit further in it. And from a QA oversight,
Mhm.
we see varying different models of QA oversight to reviewing every single batch record, right? Every single pack to order, all the way to this qualification, if you would, and Catalent providing that release. Can you speak a little bit to that?
Yeah, so where we have seen really good success to alleviate some of that overhead from a customer perspective is we take on that oversight. So we come to some agreement with the sponsor to say, right, you can audit us. All of the batch material is available for you to review and look at any time point, right? We make that very visible and accessible. But there's some agreement and qualification of our quality review, and you're providing, like in the EU, you're providing all of that documentation that's needed for QP release, country release, country approvals, all of those things that are required from that end. And essentially, we do it on your behalf. All of that is in a QTA and quality agreements that we work through, and that's part of the big, I would say, onboarding to this model in an organization is that quality oversight and how much the sponsor wants to take responsibility for, and how much overtime that they'll feel comfortable allowing Catalent to do some of the heavy lifting.
All right, great. Kate, I think you tied that nicely to the change management, right? And the organization's ability, the sponsor's ability to be able to adapt this change management throughout the organization, not just from the study managers, but through the QA team, oversight. And obviously, it's a flexible model, which I think is great, right? Okay, we have 2 minutes left. I think we've tried to address all of the questions. Bill, I think at this point, we would just say thank you to BSMA for allowing us to present here today. Thank you, Kate and Marcel, for a very interactive and informative session. And with that, Bill, I will turn it back to you, and I think we can close out.
Okay, thank you, Bonnie, Kate, Marcel, and the other Catalan folks working behind the scenes. Appreciate your support to BSMA and for all the great content here. Also, want to remind the audience that anybody who registered for this event will receive a recording of it. And lastly, to let everybody know that we have 3 more webinars that will be announced shortly that will be taking place late spring, early summer. One is on tariffs. The other is on broker selection and expectations, which is important given all the tariff challenges that many are working on. And lastly, a farmer-specific supply planning tool that's geared towards small to medium-sized biotech companies that are looking to get away from their spreadsheets and avoid full-blown oversized ERP systems. So again, more details on that, including the dates about it, you'll be hearing about over the upcoming days. So again, thanks everybody for attending, and have a good rest of the day. Thank you. Bye.
Thank you.
Thank you.
Thank you.
Bye.
Bye.